Right, now I'm going to give you a diagrammatic representation with some illustrations of the Calvin cycle. Now the Calvin cycle and the alternative mechanisms of carbon fixation. You can see the diagram here with the carbon, with the presence of ATP and NADPH, which gets consumed to get deduced or deducted to ADP and NADP plus from NADPH to give a molecule of glucose plus phosphate. And the next one you see here is the carbon cycle uses ATP and NADPH to convert carbon dioxide to sugar. The carbon cycle, like the citric acid cycle, regenerates its starting material after molecules enter and leave the cycle. Right? The cycle builds sugar from smaller molecules by using ATP and the reducing power of electrons carried by NADPH. Carbon enters the cycle in the form of carbon dioxide and leaves in the form of sugar, C6H12O6, that's glucose. So the diagram there shows carbon dioxide plus water plus sunlight gives you new plant matter and oxygen, right? Now ADP and NADPH are consumed in the process, and the carbohydrate produced is actually a 3C sugar, G3P, right? And to make one molecule of G3P, the cycle occurs 3x. That consumes 3 molecules of carbon dioxide. So note the diagram here. You could take screenshots of this on a later date once you get the videos or make copies of it. Now, the carbon cycle can be divided into 3 phases. Carbon fixation, reduction, regeneration of carbon dioxide acceptor, ρBP. Phase 1 of the carbon fixation, in the presence of carbon plus ρBP, we have rubisco, which causes the formation of 2,3-PGA, right? Each carbon dioxide, or CO2, is attached to a 5C sugar, ρBP, forming an unstable 6C sugar, which immediately splits into 2 3C molecules, that's 3 carbon molecules of 3-phosphoglycerate. This is done by the enzyme rubisco. Phase 1 carbon fixation, that's carbon from the air, with ρBP, goes to an unstable intermediate stage and splits into 3 carbon fragments, right? And then you have ribulose biphosphate and input of 3CO2 entering one at a time in the presence of rubisco, a short-lived intermediate phase where we have phase 1 carbon fixation, then we go for 3-phosphoglycerate in the presence of ADP, 6 ATP getting reduced to 6 ADP, right? That is actually the Calvin cycle part of it. Phase 2 is the reduction phase. Here each 3-phosphoglycerate receives an additional Pi from ADP, forming 1,3-biphosphoglycerate. Next, a pair of electrons from NADPH reduces the 1,3-biphosphoglycerate to G3P. And this is the formula for that. In the presence of ADP, becomes ATP with the kinase, and the end product of Calvin cycle is G3P. Right? That's a cycle there, where you have CO2 entering one at a time, plus ribulose biphosphate, that's ρBP. And in the phase 1, you have carbon fixation, where it's the short-lived intermediate phase that leads to 3-phosphoglycerate, the presence of 6 ADP to 6 ADP. You have 1,3-biphosphoglycerate, where NADPH, 6 molecules are converted, deducted to 6 NADP to give 6P. And you have glyceraldehyde 3-phosphate, G3P, which is in the phase 2 reduction, to have a G3P sugar, glucose, and other organic compounds, which is the output, right? This is the diagram for that, where you have 18 carbon atoms plus 3,5 C ρBP plus It gives 18 carbon atoms of where you have 6 G3P, given 1 G3P of sugar to be incorporated into glucose, and 5 G3Ps returned to the cycle. This is part of the process where the unstable intermediate goes to RuBP to give the stable compound. And then you have the phase 3 regeneration of RuBP. The 5 molecules of G3P are rearranged through a series of reactions to regenerate RuBP. This uses an additional 3 ADP, reason for cyclic electron flow, where you have 9 ADP and 6 NADPH, leading to a negative feedback. And then the unstable intermediate, which again the presence of RuBP and deduction of ADP to ADP and PI and NADPH to NADP in the presence of 6 X3C, that's G3P, 1 to glucose and 5 recycled, right? And then you have the input of carbon entering one at a time, the presence where phase 1 is carbon fixation in the presence of ribulose biphosphate RuBP. And then you have a short-lived intermediate, again going to 3-phosphoglycerate, with 6 atoms of ADP going to ATP going to ADP, and then 1,3-phosphoglycerate, given and 6 NADH, giving 6 NADP plus 6P, to form glyceraldehyde 3-phosphate G3P and a permanent sugar G3P sugar, right, as the output. Now, Rubisco's role is a catalyst for the fixation of carbon by RuBP. Right? Note this diagram, it should help you a lot. So the net equation for photosynthesis, 6 CO2 plus 6 H2O plus light energy in the presence of chloroplast gives 1 sugar, that's C6H12O6 glucose plus 6 oxygen, right? The alternative mechanisms of carbon fixation have evolved in hot, arid climates. There, dehydration is a problem for plants, sometimes requiring trade-offs with other metabolic processes, especially photosynthesis. On hot, dry days, plants close to matter, which conserves water but also limits photosynthesis. The closing of stomata reduces access to CO2 and causes oxygen to build up. These conditions favor a seemingly wasteful process called photorespiration. Photorespiration consumes oxygen, releases carbon dioxide into peroxisomes, generates no ADP, decreases photosynthetic output. With oxygen plus RuBP, in the presence of Rubisco, we have 1,3-PGA plus phosphoglycerate. The oxygen replaces carbon dioxide in a non-productive wasteful reaction. The oxygen outcompetes carbon dioxide for the enzyme's active site. Mechanisms of C fixation. C3 plants which combine CO2 to RuBP so that the first organic product of the cycle is a 3C molecule, 3-PGA or 3-phosphoglycerate. What we just discussed, right? Examples of rice, wheat, soybeans produce less food when the stomata close on hot, dry days. This deprives the carbon cycle of carbon dioxide and photosynthesis slows down. Excuse me. Photorespiration, an evolutionary relic. Let's see. In most plants, C3 plants, initial fixation of carbon dioxide via Rubisco forms a 3-carbon compound. And photorespiration, Rubisco adds O2, oxygen, to carbon cycle instead of carbon dioxide. And photorespiration consumes O2 and organic fuel and releases carbon dioxide without producing ATP or sugar. Photorespiration, an evolutionary relic. There's a question mark to it. Let's see. Photorespiration may be an evolutionary relic because Rubisco first evolved at a time when the atmosphere had far less oxygen and most CO2. In many plants, photorespiration is a problem because on a hot, dry day, it can drain as much as 50% of the carbon fixed by the carbon cycle. So alternative mechanisms for carbon fixation, C4 plants use an alternative mode of carbon fixation which forms a 4C compound as its first product. Examples are sugarcane, corn, grasses. The 4C product is produced in mesophyll cells. It is then exported through plasmodesmata to bundled sheet cells where the carbon cycle occurs. Has a higher affinity for carbon dioxide than Ruby P does, 2 different cells used. Once here, the 4C product releases carbon dioxide, which then enters the carbon cycle to combine with Ruby P. The enzyme Rubisco catalyzes this step. This process keeps the levels of carbon dioxide sufficiently high inside the leaf cells, even on hot, dry days when the stomata close. Right? These are the directions in the diagrams. You might see the carboxylic oxaloacetate malate to give pyruvate and carbon dioxide in the bundle sheet cell. Finally, to the vascular tissue where sugar is consumed. The 2 can plants. Stomata are open at night, closing during day. This helps prevent water loss in hot, dry climates at night. Plants take up carbon dioxide and incorporate it into organic acids, which are stored in the vacuoles until day. Then carbon dioxide is released once the light reactions have begun. The examples of these are cactus plants and pineapples. You see those? Carbon dioxide incorporated into 4 carbon organic acids, carbon fixation, and then organic acids release carbon dioxide to the carbon cycle. C4 and CAM pathways are similar in that carbon dioxide is first incorporated into organic intermediates before carbon cycle. Different in that in C4 plants, the first and second steps are separated, spatially different locations. In CAM plants, the first and second steps are separated temporarily, occur at different times. So here we have it. C4 plants events and separated in space, where mesophyll cells and the PEP carboxylate and PEP capture carbon dioxide. To convert this C4 compound to C2 compound, a bundle sheet cell, which converts in the presence of carbon dioxide, C4 compounds release carbon dioxide within the cells to give products through where we see 3PG and Ruby P and their products. And in the this is the carbon-Benson cycle, right? Here at night, the carbon dioxide is going to change to a C4 compound, which then becomes C3 to a CO2 during day. And the C4 compounds release carbon dioxide within the cells. And then you have the carbon-Benson cycle. You have Ruby P and 3BG as products. The carbon dioxide is used in the carbon-Benson cycle. So what happens to the products of photosynthesis? Oxygen consumed during cellular respiration. 50% of sugar made by plant is consumed by plant and cellular respiration. Sun is incorporated into polysaccharides, cellulose cell walls, starch storage in fruits, roots, tubers and seeds. The importance of photosynthesis. A review. The energy entering chloroplasts as sunlight gets stored as chemical energy in organic compounds. Sugar made in the chloroplasts supplies chemical energy and carbon skeletons to synthesize the organic molecules of cells. In addition to food production, photosynthesis produces the oxygen in our atmosphere. The importance of photosynthesis, a review. No process is more important than photosynthesis to the welfare of life on Earth. Thank you, photosynthesis and Rubisco. The light reactions, carbon cycle, NADPH, ADP. You have Ruby P, 3-phosphoglycerate. Here you have photosystem 2 electron transport. Chain photosystem 1, ADP, NADPH, G3P storage, starch and storage. You have the chloroplast and the starch storage giving amino acid fatty acids, sucrose as expressed. And that will be the final stage of photosynthesis done.